So I will tell you today is the Essentialome of human development and disease. So basically, when we are talking about pluripotent stem cells, and Keith gave an excellent introduction to that, we have embryonic stem cells that are coming from the blastocyst stage embryos after the fusion of the sperm and the egg, and induced pluripotent stem cells that are coming from somatic cells that by manipulation they are becoming pluripotent. And today I will also discuss haploid human embryonic stem cells that are generated only from the egg without the sperm. So basically, as you heard, human pluripotent stem cells can really make all the cells in our body. And think about that, it's a single cell that can make all the cells and the tissues in our body. And basically, when you think about the potential of human pluripotent stem cells, you can study pluripotency and human development, regenerative medicine, disease modeling, and many more. And I would like to start by really analyzing human development in a genetic way, and we'll start from the basic process in human development, which is pluripotency. So when we are thinking about pluripotency, there are different ways of looking at that. What cells are pluripotent, and those are the inner cell mass of a 1-week-old embryo. You can think about the transcriptome, meaning which genes or proteins are specifically expressed in pluripotent cells. A beautiful way of looking at pluripotency is how you can reprogram somatic cells in pluripotent stem cells. And Shinya Yamanaka showed that in a very vivid way. But today I would like to really highlight another way that we think that we can characterize pluripotency and then differentiation, and this is by the essentialome. What genes are not just expressed, but essential for pluripotency? So basically, if we want to do a genome-wide loss of function screening and see which genes are involved in this process, it's much easier to do it in haploid cells, where every chromosome is only one copy, instead of diploid cells where you have 2 alleles for each gene, so it's much more difficult to really have a genome-wide loss of function screening. So several years ago, we started from a haploid egg, and we activate that. The egg will not make more eggs, but it will divide and will differentiate into an early embryo. And amazingly, at the beginning, it's dividing and proliferating as haploid cells. Later on, some of the cells will become diploid. And through this blastocyst stage embryo that is starting from an haploid egg, we could isolate pure population of haploid human embryonic stem cells, and you can see that we characterize them by 4 different means. The most easy way to understand is that in diploid cells you have 2 sets of chromosomes, while in the haploid cells we have only 1 set of chromosomes. And by centromer staining, meaning that you are staining the chromosomes instead of having 46 chromosomes, now in each cell you have only 23 chromosomes. But you can also look by Foulkes analysis on DNA content, and again, we have half of the DNA content. So basically, that was true for undifferentiated cells that can proliferate similar to diploid cells, but what about differentiation? And we could show amazingly that we can differentiate the haploid cells, and we'll differentiate them into ectoderm, mesoderm, endoderm, the 3 embryonic lineages. So we have haploid neurons, we have haploid cardiomyocytes that also pulsating in rhythm, so they are functional, and we also have pancreatic cells that are haploid. So when you think about haploidy, you usually think about unicellular organism like the yeast. And the yeast is an excellent genetic tool, and also in my lab that we are studying human genetics, we used in the past the yeast as a genetic system. But there are haploidy in vertebrates, in multicellular organisms, and in fact, all the male ants and all the male bees are haploid. The female are diploid, but the male are haploid. But if you think about vertebrates, it's very rare to see haploidy, and you can do that only experimentally, but we have fish that are haploid as embryos, and also in rodents, and now we showed from human that we can have haploid, not only human embryonic stem cells, but also somatic cells that are haploid. So there are different ways that we are using these cells. Among them is studying what is haploidy, and we also took the haploid cells and diploid cells and generated triploid cells with 3 sets of chromosomes. This is also very relevant to human biology because in many cases, 2 sperm will get into a single egg, and you will have a triploid, 3 sets of chromosomes. And most of them will die in utero, but some of them will end in an abnormal newborn, and we could study that. But the most important thing, in my opinion, to use haploidy is for genetic screening, as this is a really very robust way of screening them. And so what we are doing, we are really mutating each gene in the human genome, and each cell has 1 mutation in 1 gene. And I'll discuss the negative selection, and later on I will discuss another strategy that I'll present later on. But basically, if we now have mutation in all the genes in the human genome, each cell has a mutation in 1 gene, we can ask whether this gene is essential for normal division or for a specific process. And so what we are looking is at the cells at the beginning and the cells, let's say, after proliferation or differentiation, and we are looking what is missing. So if the yellow cell is missing, the yellow gene is needed and is essential for this process. So I'll start with pluripotency, and we used the most comprehensive library of mutation. We are using that using CRISPR and guide RNA, and we have 180,000 guide RNA covering all protein coding genes, so it's genome-wide, more than 18,000 guide RNA genes. So basically, for each gene, we have 10 independent mutations. So if we are saying that the gene is involved in the process, it's not but 1 or 2 or 3 mutations, we have 10 different mutations. So basically, you are seeing different colors, but basically we have 180,000 different colors, and then we are looking what happened, let's say, when we are letting the cells to proliferate for a month. And then we can say which genes, once they are mutated, the cell is eliminated, and which one are taking over. So if you have that kind of an atlas of the human genome, you can study that in different ways. I'll show you very briefly 3 ways that we use that. And the first one is cell compartment. So you know that in the cell we have the extracellular or the plasma membrane. This is the outer layer. We have the cytoplasm, which is organelles, and then we have the nucleus, and another organelle is the mitochondria. And when we have done that with all of the genes in the human genome, and we could tell that there is kind of a bias from the outside to the inside. So if you are a gene or a protein that is related to the outer layer, only 1% of the proteins are essential. But if you are a protein in the nucleus and the mitochondria, between 12 to 15% are essential. And if you are cytoplasmic protein or in another organelle in the cytoplasm, it's between 5 to 7. So there is kind of these undifferentiated cells. They are really independent on the outer layer and more in what's going on in the nucleus, in the mitochondria. When I'll show you that we are really going further in development, I'll show you that this is the opposite thing. Because now we could really mutate all the genes in the human genome, we could also ask really broad questions about human disorders, because now we modeled all genetic autosomal recessive disorders. So we asked ourselves, out of the autosomal recessive disorders, those genetic disorders that have growth retardation, what percentage of them the growth retardation start at the first week of life, even before we have the different tissues. And we could show that for those autosomal recessive disorders with growth retardation, about 20% of them, we have an effect early on, even before we have the different tissues. And we could show very clearly, for example, for Fanconi anemia, there are 15 types of Fanconi anemia. All of them are showing the same phenotype. The last topic that I would like to touch regarding cell essentiality is cancer genes, and I'm now focusing only on tumor suppressor genes. And again, we could really divide the tumor suppressor genes into two groups. The one that is making most sense, once you have a mutation, the cells are growing better, and those are involved with cell death, apoptosis, and really prevent cell death once they are mutated. But those ones that are causing cancer is not causing that because they let the cells grow better. Actually, the cells without these genes goes worse. But because these genes are involved in DNA damage, the other genes that are mutated will cause the growth advantage. Regarding pluripotency, I will also show you only one slide. We are now looking at all the genes that are either specifically or enriched in human pluripotent stem cells, much more than any somatic cells, and are essential. And we see, if you think about stem cells, they have two capacities. One of them is the self-renewal, they can proliferate forever, and the other one is they are primitive cells that will make more mature cells. And now when we are looking at these genes that are essential and specific, we see these two groups. This one is the bigger group that is regulating self-renewal, and the smaller group is the one that is regulating pluripotency, or the stage of differentiation. And we could show that those two clusters of genes are regulated by two main transcription factors, the OCT4 and the E2F family of genes. And we now understand that with these two groups of genes, we can convert any somatic cell into a pluripotent cell. So I showed you what happened and which genes are now depleted, and those are the essential. But what about the enriched one? And I'll show you only one gene, and this is making sense. When we were looking at those enriched, again, they were involved in cancer and mostly in p53. p53 is the most important cancer-related genes. 50% of cancers, you can see that it is involved. And out of the 18,000 genes, the number one gene that allowed a growth advantage in our system is p53. But p53 is regulating 13 different pathways. Which one is really critical for pluripotent cells? So again, we took a genetic tool, and we could show that indeed this pathway of the IGF-1 mTOR is the most critical, and we could show that also biochemically, and we could show that they will support maintenance and growth of the cells. So this is where we induce mutations, and we show that p53 is the most mutated gene that confer growth advantage. But can we see that also spontaneously? So together with Kevin Egan from Harvard, we looked at many, many different human pluripotent stem cell lines and iPS lines, and we found that the most recurrent mutation is p53. That was actually the only mutation that we found at that time to be recurrent, but alarmingly, in cells that are really the most used in the clinic and also in research, about 20% of them were mutated. Later on, we expanded that not to look just at the p53, but on other cancer-related mutations or genes, and we found that, again, we see p53 as the most frequent, but we can see other cancer-related genes that are mutated. And when we are looking at the literature of the two most used cell lines in the literature, about 30% of them has this mutation, and the scientists at that time didn't know that they are working on mutated cells. Very recently, we expanded that to more than 2,200 samples covering about 150 different studies from all over the world, studying other undifferentiated cells or cells differentiated from them. And again, what we could see that between 20 to 30% of the cells, whether they are undifferentiated or differentiated, have mutations, again, in dark red, you can see the involvement of p53. This is the most mutated, but you can see a long list of other cancer-related mutations that exist. And the FDA now is asking investigators to look for those kind of mutations before clinical studies. So I told you a little bit about the undifferentiated or the pluripotent cells. What about differentiation? So we differentiated our cells into the 3 embryonic germ layers, the neuroectoderm, definitive endoderm, and early mesoderm, and asked what genes are essential for those type of cells that occur during gastrulation. And while in the undifferentiated cells, the most primitive cells, we could show that they have less significant essential genes in the outer layer. When you compare that to the differentiated cells, we can see for all lineages, ectoderm, mesoderm, and endoderm, that there is a more component essentiality for the outer layer, meaning that now they are getting signals from the neighboring cells that affect them. And we could really identify the signal transduction pathways that are critical for these embryonic lineages. But another thing that we want to do, and I showed you a little bit about autosomal recessive with growth retardation, but we want really to have an atlas of all genetic disorders and see for each genetic disorder at which stage during development it starts to have phenotypes. So we looked at all neurological disorders, and with microcephaly, that we know which genes are involved in this microcephaly, we could see that about 25% of microcephaly, we see an effect in very early neurons or even neural progenitor cells. So you don't have to go all the way into neurons or brain organoids to see this effect. The other 75%, you need to go further to see this effect. So we have seen that with microcephaly, and with autism, about 10% of the autism-related genes, the effect was really evident, even in neural progenitor cells, which is about 2 weeks old embryos. So I showed you that we could do the Essentialome of pluripotent cells. I showed you that we can do that in endoderm, mesoderm, and endoderm. And now we are trying to have kind of the developmental tree of different somatic cells, but now looking at the Essentialome. And we started that with neurons or caudal neurons, and I'll show you just some unpublished data. So as I showed you with neural progenitor cells, that there is a huge component to the outer layer, and the effect is really in the extracellular and the membrane. Clearly, when you compare that to the undifferentiated cells, this is also the case for neurons. And also here, with the caudal neurons, we could show which signal transductions are involved in their differentiation. So now we could really look again at neurological disorders. Now we are talking about neurons that are at the second month of life. And in addition to cephalies like microcephaly, autism, now we could look at movement disorders, hypotonia, atrophy, ataxia, and more. But we asked ourselves, what about neurodegenerative disorders? And this is very, very critical because neurodegenerative disorders, many of them develop later in life. And our cells are embryonic cells. And even if you can bring them to be fetal cells, how can you model those kinds of neurodegenerative disorders unless they have kind of a fetal phenotype? So what we showed that if we are looking at the different neurodegenerative disorders, again, it's much easier to model developmental disorders as expected, but some of them that are either early or late, both of them are adult neurodegenerative disorders, we can start to see a phenotype. So then we looked at a specific type of neurodegenerative disorders with Parkinsonism. It's not just Parkinson, but has Parkinsonism. And out of the 66 different genes and disorders, only 2 we could see early on, with neuroectoderm a little bit more, and with neurons, about 12 of them could show an effect. But again, saying that the majority, we have to differentiate them more in culture, and maybe even age them in culture. Otherwise, we won't see any phenotype. But for some of them are developmental disorders, but we can see an effect early on. So what I try to show you, that we can really look at pluripotency, what is regulating pluripotency, and what are the most important pathways in pluripotency. I didn't have to show you how we study exit from pluripotency, and then I showed you the differentiation into the 3 embryonic layers and the unpublished data regarding neurons. But we would like to really conclude with genetic disorders, and we are really trying to have an atlas of the human genome regarding human genetic disorders, but there is also a kind of an old-fashioned way of doing disorder by disorders, but we try to do that with a different angle. So many years ago, we generated the first model for human genetic disorders in human pluripotent stem cells. That was the LASH9 syndrome, and later on we did about 12 different disorders. Some of them are in ES cells, some of them in iPS cells, and with Fragile X, both in ES and iPS cells, as Keith really demonstrated very beautifully. So with ES cells, with Fragile X, we really studied the temporal regulation of the shutdown of the gene. With iPS cells, we really analyzed the neural phenotype. And using these cells, as Keith showed you, we were looking for drug screening. I will not show you the data, I discussed that, and how we are really trying to look either at drugs or food supplements to affect that. But what I would like to discuss is how we are doing genetic screening to find modifiers of Fragile X. Which genes, if they are mutated, we will not see the phenotype of Fragile X. And once we see these modifiers, can we have drug discovery through these genes and molecules that inhibits these genes? So again, what we are trying to do is now we are generate a model for a human disease in our haploid cells. And again, it's a genome-wide screening. And if the pink gene is a modifier gene, we will see that now the black circle, this is the disease, is now eliminated. And that's what we are after. And I'll just briefly show you that we really generated, in the beginning, kind of an artificial system where we introduced a transgene for Fragile X, which is unique mutation, and the gene is not expressed. And then we could show that some of the genes, once they are mutated, the gene or the marker of the gene will start to express. That really gave us a lot of data, not just on methylation, that we knew that is involved, but also on chromatin remodulators, but also recently on metabolic factors. And what we are trying to do together with Verdon and Keith is really following up on these metabolic factors and how they can affect Fragile X. So in my own lab, we are really trying to do that on all aspects of cancer research, and I showed you pluripotency differentiation, synthetic lethality and biological pathway, and also disease modeling. I showed you a little bit of that, one example. But the most important thing is the student and postdoc that really did the work. I have excellent Ph.D. student and postdocs, and Ido Sagi is the one that isolated haploid embryonic stem cells. Atil Ilmaz, again, a fantastic postdoc, together with Carmel Braverman and Shai Kainrich, is doing the Essentialome of human development at these different stages. I didn't have the time to talk with you on a different type of disorders, genetic disorders, haploinsufficiency, with Roni El Galili did a marvelous work. I showed you a little bit about the disease modeling and how we are looking for modifiers. There's the work of Dan Vershkov, and I didn't have time to discuss the work of Shiran Bar. And regarding the spontaneous mutation, I showed you a little bit of the work of Eliad Lesmi and Jonathan Jung, and you heard Keith, and it's a pleasure to collaborate with him on Fragile X and carry that to the next stage together with Vernon Taylor from the University of Basel. Thank you.